Hello, dear students. Previously, we have discussed that ATP is the energy currency of life, and usually ATP provides energy by hydrolytic reactions. So, today we will discuss that ATP provides energy not always by hydrolytic reactions but also by group transfer reactions. So, today we will discuss that whenever ATP provides energy, it is not always by hydrolytic reactions. In some situations, this is the case, but this is not the case in all reactions. Sometimes it will occur by hydrolysis, and in other cases, it will occur by group transfer reactions. So, throughout biochemistry, whenever we encounter the reactions in which we see that ATP is providing energy for the conversion of the reactants to the products, we will see that this is represented by one single arrow. And this usually shows that ATP is converted into ADP and PI, meaning adenosine diphosphate and inorganic phosphorus. Whenever it is written in this way, it gives a general impression that the reaction has occurred by means of hydrolysis. So let's take the example of conversion of glutamate to glutamine. The process is catalyzed by the enzyme glutamine synthase, and here it is represented by a single arrow. So it appears that the reaction goes in a way that ammonia attacks glutamate, and in the presence of ATP, the phosphate bonds are broken, the amine group is attached, and it causes the formation of glutamine. Whereas in nature, this is not the case. So in nature, it is a two-step process, and it occurs in a way that in the presence of glutamine synthetase enzyme and ATP, glutamate forms a covalent bond with inorganic phosphorus. We can see that the phosphoryl group is not leaving the medium, and in the second step, this phosphoryl group is replaced by amine group, and it causes the formation of glutamine. So this process has taken place via the group transfer mechanism. There are some biological reactions which take energy by the hydrolysis of ATP. So these include the non-covalent binding of ATP or other nucleotides like GTP, followed by the hydrolysis causing the release of ADP or GTP and a phosphoryl group. And the examples of this kind of reaction are muscle contraction or cycling of proteins between two different conformations like in tautomerization or so. The second example is the movement of enzymes along DNA or the movement of ribosomes along the messenger RNA, or the energy-dependent reactions which are catalyzed by helicase enzymes or some topoisomerases, and the processes which are carried out by the GTP-binding proteins that act in signaling pathways and play their role in the conformational changes. So we discussed before that the phosphate compounds found in living organisms can be divided into two groups based on the standard free energy changes as a result of hydrolysis. So one is high energy compound and the other is low energy compound, and we discussed the details before. Here an important point is that usually the term high energy phosphate bond is used by the biochemists to describe the bond between phosphorus and oxygen, meaning it is usually said that this is a high energy phosphate bond. This is broken in hydrolysis reactions, and because of the breakage of bond, the energy is released. So the fact is that this is giving a kind of wrong impression, and it gives the impression that bond itself is containing some energy. But the fact is that the breaking of all of the chemical bonds would require the input of energy, whether it is the oxygen to phosphorus bond or whether it is any other bond. So another point is that the free energy changes in the sequential reactions are additive to each other, meaning if so for example we can look at that PEP, meaning phosphoenol pyruvate, is being converted to pyruvate and inorganic phosphorus or pyruvate and ATP. So this is the final step of the reaction, and this final step proceeds via these two steps. And the fact is that the energy which will be produced in this reaction and the energy which will be produced or consumed in the other reaction, they are all additive, meaning we can add all this. So in the first step, PEP is converted into pyruvate and inorganic phosphorus and releases 61.9 kilojoule energy, whereas in the next step ADP and inorganic phosphorus are converted into ATP and water with the input of energy, meaning it has consumed some energy. And the final step is that PEP plus ADP has generated pyruvate and ADP. And here we can add these two steps, the energy which is released or consumed in these steps, and this is the final amount of energy which is released during the whole reaction. So although for the above reaction we have calculated the free energy change just like a normal algebraic addition or subtraction, but maybe you have noticed that in third reaction you can see there is no release of inorganic phosphorus. So this indicates that PEP has directly donated its phosphoryl group to adenosine diphosphate. Another point is that on the basis of the standard free energy changes of hydrolysis, the phosphorylated compounds are categorized as having low or high phosphoryl group transfer potential, which is also represented as delta Gp. It was discussed in previous lecture. And the fact is that the transfer of a phosphoryl compound effectively puts energy in the compound to which it is donated, and this energy can be used in the subsequent metabolic transformations whichever will be carried out by this compound.